everybody, I am Deepika from LearnoHub, a free learning platform where you can study physics, chemistry, maths, biology absolutely for free only at learnohub.com. So students, we are going to start with our ICSE class 10 biology with the chapter number 2, that is structure of chromosome, cell cycle and cell division. So let us start with the chapter. So starting with the first topic of the chapter, that is cell division. So cell division is the basic and important characteristic of life. It is the fundamental characteristic of life. Now, as we see generations after generations, so this is possible only because of cell division. When the cell divides, the life continues. And even if we talk about the simplest cell, say amoeba, to coming on to the complex animals, say humans, say a big giant elephant, say a tiger, say a blue whale, and from the simple small plant to a tall and a complex plant, every living organism is made up of cell, and cell division is the fundamental characteristic of life in all these organisms. So all the easily noticeable events that happen during cell division are all related to chromosomes. So these chromosomes, they get duplicated and they are passed on to the daughter cells, and this is how a living organism's body consists of the same chromosomes throughout their cells. So the very important question over here is how this cell division happens, but before that we will discuss what are these chromosomes, because these chromosomes are being duplicated and they are being transferred to each and every cell of the body. So let us start with our next topic, that is chromosomes. Talking about chromosomes, so when we talk about a cell contains a nucleus, right? So when this cell is not stained, when it is a unstained cell, in that case there are many components inside the nucleus which are not visible, which cannot be noticed. But when this cell is stained with a particular dye, with a suitable dye, in that case the contents of this nucleus, they start becoming visible. So what does a nucleus contain? Nucleus contain the most of the cell's DNA, and it is organized into discrete units. Fine. These chromosomes, they are long molecules which have associated proteins with them. And what are these proteins called? These proteins are called as histones, right? And together this DNA and these proteins, they are called as chromatin. So what is chromatin? DNA and proteins together are called as chromatin. Now, so there are different stages of the cell. Sometimes the cell is dividing, sometimes the cell is not dividing. But at the time when cell is not dividing, it is in the non-dividing stage. At that point of time, these chromatins, they appear to be like long thread-like structures which are tangled amongst themselves. So just think about a bowl having noodles, right? So how do those noodles look? They are tangled inside each other and they are the long thread like structures, right? So same is the case with the chromatin. So in the non-dividing stage, they look like a long tangled mass of thread. Okay, so this is a cell, right? This is the nucleus and these are the chromatin fibers inside the nucleus. So these are extremely thin and these are darkly stained in a non-dividing stage, right? Now coming on to when cell starts dividing, so what happens at that point of time? So at that point of time, these chromatin material, they start to condense, they start to thicken up and they start to condense and they form a particular structure which looks like this. And what are these called? These are called as chromosomes. Fine. So simple. This is the non-dividing stage, right? And this is the dividing stage, where the chromosomes are clearly visible. When this chromatin material becomes condensed, it becomes thick, and they start picking up that dye with which this cell was stained, and now we can easily see these chromosomes. Fine. So I hope this is clear. So what are these chromosomes? These chromosomes are highly coiled, condensed chromatin fibers. What are these? Highly coiled and condensed chromatin fibers. So coming on to the discovery of the chromosomes. Who discovered chromosomes? Chromosomes were discovered by a German scientist, Walther Fleming, in 1882, and he discovered these chromosomes in animals. Now talking about where did he find these chromosomes? So he found these chromosomes in the larvae of the salamanders. So at this point of time, the cells were in the dividing stage, and he saw these chromosomes to divide in a longitudinal manner, in a lengthwise manner, right? So these cells were dividing, and he called this division as mitosis, which actually means thread, right? So because these were the long thread-like structures which were dividing, so he used the term mitosis for this type of division. So he is the man who discovered chromosomes. Now when he was discovering these chromosomes, when he was studying these cells, he was studying these chromosomes, at that point of time he only had a very simple, a very old type of a microscope which could not study those details in the chromosomes. But as this microscopy it got advanced, these chromosomes were studied in greater detail, and more details were observed in these chromosomes. Now talking about chromatin, we already spoke about chromatin when we were discussing about chromosomes, right? So what are these chromatins? These chromatins are made up of two things. One is DNA and the another one is histones, which are proteins, right? So this chromatin material is made up of DNA, that is deoxyribonucleic acid, which is around 40%, which constitutes around 40% in these chromosomes, and then coming on to histones, which are proteins, and they constitute around 60%. So this is the way a chromosomes look like when they are in the dividing stage, right? And basically what is there inside these chromosomes? So these are the thread like structures, highly tangled mass of threads which appear like this, right? So there are DNA molecules inside these chromosomes, and these DNA molecules, they replicate, they make messenger RNA, and then these only make what? Proteins, right? Proteins which are used by our body. So this is how, this is a chromosomes, this is the chromatin material which is shown in this picture, and this is the basically the more detailed part of this chromatin is this. And what are these? These are the histones, right? So what are these? These are the histones. Histones are what? Proteins. Okay, so there is a very important role of these histones during the cell division and many other processes. So we will study them in the further discussion. Okay, so I hope this is clear. Let us see our next topic. Now talking about the scientists who worked on the molecular structure of DNA. So this all started in the year 1953, where Rosalind Franklin, she studied the shape of DNA molecule. So she was the first who studied the shape of the DNA molecule in 1953, and in the same year there were two more scientists, namely Watson and Crick. They tried to make the structure of the DNA, and for which they received a Nobel Prize in the year 1962. So Watson and Crick, they worked on the structure of the DNA, and they proposed a double helical stranded structure of DNA and received a Nobel Prize. So they were who gave this double helical structure of DNA. So it was Watson and Crick, right? So I hope you will remember these names. These are some of the important names while we study about the DNA molecules. Now, talking about the molecular structure of DNA, a molecule of a DNA is very huge, it is very large, and that is why we call it as a macromolecule. Now, there are two strands which are bound to each other. So, this is the strand 1, right? So, this is the first strand, and now this is the another strand, right? So these are the two strands which are bound to each other, and there are repeated nucleotides. And what are these nucleotides? So there are the three components of the nucleotides. So these three components are a pentose sugar, a phosphate group, and nitrogenous base, right? So let us talk about these nucleotides in detail. Coming on to the next part of the topic, that is nucleotides. So what are these nucleotides? So just now we studied there are two strands in a DNA molecule, right? So these two strands, they are made up of what? Nucleotides, right? The repeating nucleotides. And what are these repeating nucleotides? So these nucleotides have three components. One is phosphate, then sugar, and then finally we have some bases, right? So there are basically four bases. So these two strands, they are wound around each other, and this is how they look. So what is there on these sides? So on the sides are phosphate and the pentose sugar, and inside, joining these two strands, are the bases, right? So how these bases are joined, that we will study, but this is how it looks. So on the side is this phosphate group, then is this pentose sugar, right? And inside too is the base. So more clarity you will get from this picture, that is B picture. So these this is the phosphate, this is the sugar, and this is the base of this strand, and this is the base of this strand, right? So again there is a phosphate and there is a sugar, pentose sugar, and this is the base. So this is how these two bases, they are joined to each other with the help of hydrogen bonds. So now what do they make? They make a ladder like arrangement, right? Can you see this picture? So these are the sides of the ladder, and these are the rungs of the ladder. Rungs of the ladder means these are the steps of the ladder. So it looks like something like this, right? So now let us discuss in detail about the bases that are present in these DNA molecules. So talking about the nitrogenous bases in DNA. So in DNA there are four nitrogenous bases: adenine, thymine, guanine, and cytosine. So A for adenine, T for thymine, G for guanine, and C for cytosine. So always remember, adenine and thymine, they pair with each other. So on the one strand there will be adenine, and on the complementary strand there will be thymine, and these two nitrogenous bases, they pair with each other with a double bond, right? So there are two hydrogen bonds that are present. Now coming on to the next two, that is guanine and cytosine. So guanine and cytosine, they always pair with each other, and there are three hydrogen bonds between them. So this is how the pairing happens. So you can see the DNA molecule besides this text. So these are the two strands, right? So what is there on the strand? So there is a phosphate group, and there are sugar molecule, that is pentose sugar, right? And with this sugar is attached these bases. So this is guanine on the first strand, and on the second strand is cytosine. So these guanine and cytosine are pairing with each other. In the same way, you can see here it is adenine on this strand, on say on the strand 2, and on the strand 1 there is thymine. So adenine and thymine, they are pairing with each other. So between adenine and thymine, two bonds, and between guanine and cytosine there are three hydrogen bonds. Clear? So I hope this is clear to everyone. This is a very simple concept. So how these two DNA molecules are bound to each other, why they always stay together with each other. So let us see what is next. Now talking about histone proteins. So we studied about histones when we were talking about chromatin. So what is a chromatin? The DNA and proteins together make up chromatin, right? So now talking about histones. So histones are nothing else but they are just the proteins which help in coiling and packaging of DNA into the structural units which are called as nucleosomes. So there are eight histones which together combine together, which join together, and what do they form? So these are 4, 5, 6, 7, and 8. So these 8 histones, so this is histone octamer, and this octamer has this DNA molecule which is coiled around this, and it helps in packaging of this DNA. Now we know that a human nucleus is just 6 micrometer, right? So it is very small. But if we talk about a chromatin, or the approximate length of the DNA molecule, so it is around 2 meters. So the DNA molecule is around 2 meters in case of a human cell. A single human cell has a nucleus, and in that nucleus there is around 2 meters of DNA, but that nucleus is just 6 micrometer. So we need something that should just fit and coil and just pack this DNA molecule so that we can fit it inside that nucleus. So what are those? Those are the histone proteins. So a single human chromosomes may have about millions of nucleosomes. So there are millions and millions of nucleosomes inside a single human chromosomes. So this DNA basically it needs to be condensed, right, so that we can fit it inside the nucleus. Now each chromatin is coiled, it is super duper coiled, and it forms chromosomes. So do you remember how right now we all just use these mobile phones, right? But some of you might have those corded phones at home, right? So they have what, the cord is always attached to the main set with a coiled structure, with a coiled wire. So that coiled wire is nothing else but it is a chromosomes which are coiled around these histone proteins. Okay, so take a ball and just coil a thread. Okay, so that will tell us how these nucleosomes will look like. So these octamer, this histone octamer having this coiled DNA will be called as nucleosomes. Okay, so let us now talk about further details. So let us now summarize all the concepts that we discussed till now. So do you remember when we were studying chromatin? So I told you just consider a bowl full of Maggi noodles, which is favorite of many, not all, but yes, many. So this is how that bowl of noodles look like, right? So in my case, these noodles are green in color because I love green color and I love that greenery around me. So I love plants, right? So nothing to do with this chapter, but still. So this is a bowl full of these Maggi noodles, right? So when we see a cell under a microscope in a non-dividing stage, so this is how those chromatin fibers look like. So what are these? These are basically the chromatin, right, inside the nucleus. So in the same way, this is the wall of the nucleus, these are the nucleus pores, and these are the chromatins. So what are these chromatins? DNA plus histones, right? And from if we enlarge these structures, this chromatin structure, we will see something thread-like structures, and inside that thread-like structures, when we enlarge it a little bit more, what we will get? We will get these histones, these circular kind, these ball shaped structures which are histone proteins, and these thread like structures which are nothing other than DNA. So these are DNA, right? And these are histones, so already marked in this, right? Now when we elaborate, or when we zoom a little bit more into the structure, so this is how we will see this DNA structure. So these are the DNA structure, this is the double helix, and inside are these small histone proteins which are circular in shape, right? And when the cell is in dividing stage, this is how we see it, a condensed chromosome, right? So we see it in a very condensed manner, and very clear picture of these chromosomes we get when the cell is in the dividing phase. Let us now dive more into the detail and just discuss this everything that we studied. Now talking about the summary of what we have studied till now. So we will summarize everything in the form of a flowchart. So this is what we have studied till now. So what are these called? These are called as chromosomes. And what are these chromosomes made up of? The chromatids, right? And what are these chromatids made up of? DNA plus histones, right? So if we pull out a thread from this chromosome, it will come out like this. And what is there inside? So it is DNA and histones. How many histones? 8 histones. So it is a histone octamer. Now zooming into more detail of this structure, we will see something like this, the strands of DNA. So the strands of DNA are made up of phosphate and pentose sugar, and these two strands are joined with each other with the help of nitrogenous bases, which are joined to each other with the help of what? Hydrogen bonds. So adenine pairs with thymine and guanine pairs with cytosine. So same are the number of bonds. Like adenine and thymine, so there are 2 hydrogen bonds, and in case of guanine and cytosine, there are 3 hydrogen bonds. Now what is it? This is a DNA, right? So DNA is surrounded by a core of 8 histones, which together forms what? Nucleosomes. Now seeing this, what is it? This is a nucleosome. So these are the 8 what? These are the 8 histone proteins, and coiled around it is DNA. Now these are the coiled structure, these are the super coiled structures, and these are the chromatin fibers, these are the chromosomes, and these chromosomes are present inside the nucleus. This is the nucleus, and where is this nucleus present? Inside the cell. Fine. So this is the summary of what we have studied so far. Now talking about the structure of chromosomes. So when we talk about the structure of chromosomes, so the chromosome something looks like this, right? So this chromosome has 2 sister chromatids. So when we draw a structure of a chromosome while we are talking about cell division, so we draw it something like this, right? And this point where these 2 sister chromatids they meet, these are called as, this is called as centromere, right? And these 2 are called as sister chromatids, right? So these are the arms of the sister chromatids, right? And these are the sister chromatids, these are the arms, right? And this is what? This is nothing other than but a centromere, a center which joins these 2 sister chromatids. Now this centromere plays a very important role when we are talking about cell division, because when cell division happens in case of animal cells, so this centromere is a place where the spindle fibers attach, because when you will study about the cell cycle, cell division, how it is happening, so the different stages of cell division you will find. So first these are just like, these chromosomes are just like the chromatins, like the Maggie noodles, like a bunch of thread, a thin long thread which are just coiled around each other. Then they will condense and they will look like this, and after that there will stages will come when this chromosome will split. So it will like split into half. This half portion will go into the one side of the cell, and the other half will go to the other side of the cell, right? Other side of the nucleus, right? So this is what is called as a sister chromatid, right? These are the sister chromatids, these are the centromeres, and centromeres is the place where spindle fibers attach, and they just pull this this chromosome and divide it into two. So after the completion of the cell division, these chromatids, they decondense, right? So they will exactly look like those long thin condensed fibers they used to look when they were in the non-dividing stage. So once the cell division is complete, again they will go to that non-dividing stage where they will decondense, they will convert it, they will convert them into a long thin thread like chromatin fibers, right? So this is the structure of a chromosome. All right. Now talking about genes. So genes, they are the specific sequence of nucleotides. So there is a chromosome, right? There is a chromosome. On this chromosome, this is made up of DNA, right? This chromosome is made up of DNA. So there are DNA sequences. So these DNA sequences, let us consider this is a double helical DNA, right? So there are the two strands of DNA. On this strand of DNA, there are specific nucleotides which encode for a particular protein. So as you go to your higher classes, you will understand that whole say the stretch of DNA is not useful, or you can say does not code, but there are certain segments of DNA that codes, and we call them as exons, right? What do we call them as? And the others are called as introns. So these are the specific sequences of nucleotides which encode for a particular protein and which express in form of some particular features in the body, right? So this is a DNA sequence. Just say this is the sequence of nucleotide that encode for eye color, right? So this particular portion will form certain protein. First it will be converted to messenger RNA, then it will form proteins, and these proteins will encode for a particular color of eye, right? So this is how genes work. Now these are the units of heredity. Now we have heard that characters, they pass on from generation to generation. Like from your parents you must have inherited few characters, like might be your eyes look like your mother, your hair are on your father. So all, from where do these things come from? So these are the genes which are responsible for heredity. So these genes have been passed from your parents to you, and that is why your eye color, your hair looks like that, right? So they are responsible for specific characteristics in offsprings, right? So I hope this is clear. Let us see what is next. Now the question is, why there is a need of new cells? Why do we need these new cells? So these new cells are required for various purposes, right? So the first thing is growth. So if we want to grow, if our body wants to grow in size, so that ways we need what? We need new cells that keep on adding to our body, right? So remember when in previous classes you must have studied about reproduction. So in reproduction, what do we learn? We learn that there are two cells, two gametes. So one is sperm cell and another is the egg cell, right? So these two cells, they fuse together, right? So they fuse together and they form a zygote, right? So what is it? This is sperm and this is egg cell, right? And when these two fuse, when this egg is fertilized by sperm, it forms zygote, right? So now what happens? So this is a single cell. On the fusion we got a single cell, and now this single cell will start dividing and it will keep on multiplying, and then it will form millions of cells, and this is how the organism will grow, right? Now this happens with almost every living organism. Every living organism, whether it is a plant, whether it is animal, so every living organism starts its journey from a single cell, and as the time grows, as the cell divides, the size of the organism starts growing. So do you think we are made up of a single cell, such a huge single cell? No, there are millions and billions of cells which make up a human being. So now what happens, these cells keep on dividing, and on dividing they form a cluster, right? So the cell starts forming a cluster. So these clusters, when the cell forms a cluster, it forms tissues. Now these tissues, when combined together, right, they perform specific functions, right? So what do they form? They form organs, different organs of the body, say liver, say some other organs, say heart, say lungs, right? Now there are different organs which together make up organ system. Now organs together combine together and they form organ system. All right, so now what is happening? So life is starting from a single cell, and these cells are clustering together and performing various functions that are important for life. If you come to alimentary canal, right? So our digestive system, our alimentary canal is composed of many cells. Now in this case, what happens, there is a mouth, there is esophagus, there is stomach, there is intestine, there is rectum, right? So all these are different organs which have together make up one organ system. So this is why we need new cells. So now as the organism grow, these cells cluster and form a organ system, and these organs, they perform their function, and this keeps the life going, right? So cell division is very essential for growth. Now coming on to the second point, why do we need new cells? So new cells are required for growth. And now the second point is replacement. Now we know that there are millions of cells in our body, and obviously during our normal functioning, these cells suffer some damage, and these damaged cells should be replaced by the new cells, right? So see, there are millions of cells every day. There are many cells that get destroyed. Now if these destroyed cells do not get replaced, so do you think our body will be able to function properly? So the answer is no. So the normal wear and tear of the cell, it happens, like for example, a very easy and very good example, that is around 2 million RBCs are destroyed every second in our body. Now see, RBCs are very important to us. Why? Because they carry hemoglobin. They have hemoglobin which carries oxygen, and in turn they carry carbon dioxide which is thrown out of the body, right? So now see how important are these RBCs to us. So if they do not get replaced, so do you think we will be able to survive? So every second around 2 millions of RBCs are being destroyed. So now they are replaced with the new cells in the bone marrow. Now coming on to plants, the same happens in the plants. The old and the dried cells, old leaves, they fall off. So do you think once the leaves fall off, the new leaves do not grow? No, the new leaves do grow. And how do they grow? A cell keeps on dividing and dividing, and this is how we get new leaves from on the trees, right? So how is this happening? With the help of cell division. Now the point number 3, we need cells for repair too. Now sometimes what happens, while working in the kitchen, or while performing some experiment, or while doing some activity, you might get that cut on your skin, right? And sometimes while playing, someone might get fractured, might be his hand, might be his leg, might be his arm, right? So on all those cases, what do we need? We need cells to divide. right? When the cells will divide, these broken cells will get replaced, these gaps will get filled only when there is cell division. Okay, so we need cells for repair too. So talking about the fourth point, that is reproduction. So if we talk about amoeba, if we talk about bacteria, a cat, a dog, a banyan tree, say a small plant, a rose plant, so every living organism starts its life from a single cell. Many of them remain as a single cell, but many of them, they grow into millions of cells. So let us talk about amoeba first. So what happens in case of amoeba? So this is amoeba, right? So this amoeba will divide and it will form two more cells, right, of the same kind, of its own kind. So this is the parent cell, and these are what? These are the daughter cells, right? So with the help of the cell division type, that is mitosis, this parent cell will divide into two and will produce two independent cells that are called as daughter cells. Why independent cells? Because this single cell is able to perform all its functions that are important for living on its own. This cell does not need any other cell to perform its life processes. So now coming on to say humans, a banyan tree, so there are reproductive organs. So what happens in these reproductive organs? These reproductive organs produce some gametes, right? A male gamete and a female gamete, and with the help of these gametes they reproduce. Now what happens in case of humans? So very interesting to and easy to understand is the case of humans. So what happens in case of human males? So we show it like this, and there are females, right? So we mark it like this. So there are how many chromosomes? There are 46 chromosomes. So that means there are 23 pairs of chromosomes. How many pairs? 23 pairs of chromosomes. Now what happens in these cells? They produce gametes, and these gametes are, they have 23 chromosomes each. It means what is happening over here? Meiosis. There is reductional division. The chromosome number becomes half. From 46 it turns into how much? 23. Now what will happen during fertilization? So there will be a sperm, right, and there will be an egg cell. Now when sperm will fertilize this egg cell, so 23 plus 23 will be equal to 46. So it means what they will form? They will form a zygote when they will fuse with each other, and the number of chromosomes at that point of time will be 46. So this complete cell will yes, the zygote is a single cell, and life starts from this single cell, that is zygote, and it has 46 chromosomes, that is 23 pairs of chromosomes, right? So this is how it happens. So in case of reproduction also, we need cell division. Now this zygote, say this is a zygote, so now this zygote will start dividing, and this single cell will grow into millions and billions of cells and produce a single organism, right? So now can you imagine how important is this cell division, and how important is the production of new cells in our body? If new cells, our body stops producing new cells, we won't be able to grow, we won't be able to repair our body, we won't be able to replace the damaged cells in our body, and also we will not be able to reproduce, right? So life will not be possible without cell division. Now coming on to the important part of the chapter, that is cell cycle. What is cell cycle? Cell cycle is just divide, grow and redivide, right? As it is the heading of this topic, divide, grow and redivide. So what happens? First a cell divides, it grows into a certain amount, say it duplicates its internal things, say DNA, proteins, into a certain amount that is required, and then it again divides, right? So that is cell cycle. So what happens? A cell grows in size, right? It synthesizes the materials like proteins, RNA, and duplicates its DNA, and then finally when it has the enough amount of these proteins, RNA, this DNA, then this cell will split and form the two daughter cells, right? The cell will grow, then it will split into two daughter cells, and now what will happen? These two daughter cells will further grow to a certain size, duplicates all the contents that are required like proteins, RNA and DNA, and further they will also grow in size and they will redivide, right? So this is cell cycle. So it is an organized and predictable series of events which are performed by cell to divide. So there are some organized and predictable series of events that are performed by a cell, right? So what are these organized and predictable series of events? That is what we are going to study in cell cycle, on the different phases of the cell cycle: interphase, prophase, metaphase, and all those phases, right? So this cell cycle is very organized, it is very predictable, and all the things happen, and it happens in all the cells while they grow and they divide, right? So there are two phases of the cell cycle. One is a non-dividing phase and another is dividing phase. What is this non-dividing phase? Non-dividing phase is interphase, right? And what is this dividing phase? It is M phase, that is mitosis. Okay, so I hope this much is clear. Now when we talk about mitosis, so mitosis happen in almost all the cells of the body. So what happens when a cell, when we need to repair a cell, when we need to replace a cell, when our body needs to grow in size, so in all types of cell divisions, mitosis happens. So the chromosome number is 46 and it remains 46 when the cell grows and divides. But only in case of gamete formation. We just studied about what? We just studied about the human gametes, how they are formed, where they are formed, right? So what happens in case of gamete formation? The chromosome number reduces to 23, and except that everywhere the mitosis happens, and only in case of gamete formation, which type of cell division happens? So that is meiosis. So what is meiosis? It reduces the number of chromosomes to half. So we call it reductional, D for division, right? And for mitosis, what do we call it? So it keeps the chromosome number equal. So that is why we call it as equational division. Coming on to the first phase of cell cycle, that is interphase, and a very, very important phase. Now, what happens when a cell divides? So the mother cell and the daughter cells, if we compare the size of a mother cell and the daughter cells, so the daughter cells are very small in size as compared to the mother cell, right? So this is the mother cell, this is the fully grown cell to its maximum size, and now the daughter cells produced be somewhat like this, little smaller than the mother cell. Now the cell prepares during this phase, during interphase. Now these cells will prepare themselves for the next division. So they need to actually do some activities inside the cell. That is this is the way how they will prepare for the next division. Now first of all, the cell grows in size as the mother cells, right? So these daughter cells will grow in size as the mother cell, and it feels like there is nothing else going on inside the cell. The cell seems to be little in a, say, resting phase. That is why initially this phase was called as the resting phase, because it was thought that there is nothing that was happening inside the cell. But very, very important things happen during this phase of cell cycle. So there were no visible changes, but the cell is actually quite active and it synthesizes more and more of the DNA. So there are basically the three important phases that we are going to study under interphase. The first one is first growth phase, and we call it as G1 phase, right? So it is first growth phase, right? Then comes the next phase, that is S phase. We call it as synthesis phase, right? And now the third one, that is G2 phase, and we call it as second growth phase, right? So first phase, synthesis phase, and the second growth phase. Now if we look at all these phases, so after G2 phase there comes the mitosis. So if you look at this picture, it is very much clear that this is the interphase. So it is from here to here. So basically it starts from here and ends till here. But if you see, this interphase is the longest phase in the cell cycle. So let us understand what happens in each of these three phases of interphase. Now the first phase of interphase, that is first growth phase, and we also call it as G1 phase, right? So what all happens during this G1 phase? So RNA and proteins are synthesized during G1 phase of interphase, and also the cytoplasm volume increases, right? So initially when a cell divides, so this is a mother cell. From this we will get two daughter cells. So here the size of the nucleus is fully grown. This nucleus is actually fully grown, but the cell cytoplasm is not that grown, right? Is not in the amount that a cell needs to divide, right? So in this case what happens, now the cytoplasm volume will also increase. So when the cytoplasm volume will increase, eventually the cell will grow in size, right? Also in case of animal cells, mitochondria will start dividing, and in case of plant cells, chloroplast will start dividing. Why? Because they have their own DNA. That is why these two organelles, they start dividing in animal cell and in plant cell respectively. Now later what happens in G1 phase, either the cell will withdraw from this phase, from the cell cycle. So either what will happen later, either the cell will go to G0 phase, and we call it as a R phase or resting phase or G0 phase. Some in some books you will also find it as G0 phase. So either cell will enter the cell cycle, right, or they will withdraw from the cell cycle and they will enter the resting phase, that is the R phase or the G0 phase. And if the cell wants to enter the cell cycle, so it starts entering the next phase of this interphase, that is the S phase, and that is the second phase of this cell cycle, I mean the interphase, right? So if you see, the length of the G1 phase is more as compared to the S and the G2 phase. Now coming on to the next phase, that is synthesis phase, or in short S phase. So S phase is the point when DNA starts synthesizing, or the DNA duplication starts. So this is the phase of DNA replication. Now this is the phase where chromosomes get duplicated. Obviously, where is this DNA present? So what is this chromosome made of? It is made of chromatins. And what is this chromatin made of? DNA and histones, right? So this is how this chromosome is also getting duplicated, but it still remains attached. These like segregated chromosomes we cannot see in this phase. So which phase is like during which phase DNA replication happens? So it happens during S phase. Coming on to the next phase, that is second growth phase, which is also called as G2 phase. Now talking about G2 phase, so this phase is the shortest of all. And now just recall the first growth phase. What all happened in the first growth phase? So there was synthesis of RNA and proteins. Then the cytoplasm also increased, so which increased the size of the cell, right? When we came to the second phase, that was the S phase, that time DNA was replicated. So the DNA got duplicated, right? Now coming on to the third phase, that is second growth phase, G2 phase. Now this phase is the shortest of all. here the RNA and proteins which are left, they are synthesized and the cell is now ready to undergo what? Undergo cell division, and the cell then enters the dividing phase. So in this phase, so what happens? This is the M phase, right? The mitosis that happened. Now after M phase, now there is a mother cell, right? This divides into what? Two daughter cells. But if you see the size of the daughter cells as compared to the mother cell, they are very small, because the nucleus is fully grown but the cytoplasm is still very less, so it is not a fully grown cell. Now when we come to then the cell enters this G1 phase, the first growth phase. That time cell produces some protein, some RNA, and now at this point, at the last the final points of G1 phase, the cell decides whether this cell has to enter the next phase, that is the S phase, that the cell wants to divide or not, or it wants to go into the resting phase, right? So either it will go into the R phase, resting phase, or I told you sometimes it is said to be as G naught phase. Now after this, the cell will go to S phase, then G2 phase. Now when the cell enters the G2 phase, or we can say when the cell enters S phase, this means now this cell is going to divide and completes all the phases of cell cycle. Now after the G2 phase is over, what will happen? The cell will now enter the M phase, that is the mitosis, right? Now coming on to the formation of new DNA, right? So where did this new DNA starts forming? So it started in S phase. So what happened in S phase? The DNA replicated. So what happens, how it happens, not the detailed part, but the simple part that we will discuss now. So this happens in the S phase of interphase. Then this DNA molecule duplicates so that when the cell will divide into the daughter cells, both the daughter cells will have equal amount of what? DNA, right? So the DNA molecule duplicates for equal distribution of two daughter cells. DNA double helix open at one end, strand becomes free, right? So this is the DNA molecule, right? This is the parent DNA molecule. Now what will happen? This DNA will start unwinding. The hydrogen bonds between the between two strands will start breaking. So it will unwind, it will open, and then what will happen? The DNA synthesis starts from the opposite ends, right? So can you see now the DNA, this is the new DNA strand that has started forming, right? Now this will continue, and finally there will be two DNA strands which are made from this parent DNA. So what are these? These are the daughter strands, right? Easy and simple. So this process is actually complicated. There are many things that are involved in this. There are many enzymes that are being used during this DNA replication, but that is a little complicated process. Not very complicated once you understand it nicely, but we need to give some time to understand this, because this is a concept that we will study in our higher classes. Now the question is, can the cell cycle go endlessly? Can you think of a cell division that will go endlessly? So the answer is no. So at some places the cell will stop dividing permanently. At some it will stop dividing temporarily, and at the other point, at the other place, it may stop dividing until it is not needed, right? So let us understand those different examples of all these scenarios. So there is always a regulatory mechanism which regulates the body functions, which regulates this division also. So now there is a regulatory mechanism for cell cycle also. Now the brain cells or the nerve cells or these kinds of cells, they just grow. I mean, the nervous tissues, or you can say the cells in this nervous tissue, they just grow, or they just divide, or they are formed only at the embryonic stage, when we are in the womb of a mother, right? So at that time, all the brain cells and the nervous cells have been formed, and now they do not divide further, so they are not even replaced. If a nervous cell dies inside the body, right, so that nervous cell will always be dead, and it will never be replaced throughout our lifetime. Now coming on to the liver cells, if we talk about liver cells, so they are replaced but once or twice in a year or in 2 years. So that's it. They are not replaced regularly. Like we are talking about skin. So skin cells, they keep on our body keep on replacing these cells regularly, but not the liver cells. So once or twice in 1 or 2 years. Then coming on to surface skin cells, so they are being replaced. Now what happens, because this is the most exposed part of our body, now the skin cells, they get damaged, right? So our body needs to repair them very frequently, or you can say, or on the regular basis, or continuously, so that our internal parts are safe, right? Now if you see around you, there is a lot of dust, there is see, when we rub our skin, when we scrub our skin, so on all those points, the skin cells, they keep on shedding, and the dust even constitutes our skin cells. The dust that we broom, that also constitute our skin cells, so they are being replaced continuously. Then comes to plants, so the same happens in plants. So there are meristems are the points where the cell has those meristematic tissues which are active in division, like in case of a bud, in case of the shoots, in case of the root tips, shoot tips. So in all those cases, there are meristems, the meristematic tissues which keep on continuously dividing. So these cells are being, they are being continuously dividing to increase the length of the plant, to replace the cells, right? Then coming on to the germinal cells, so these are the specialized cells in the ovary, in the anther or the testes. So they undergo meiosis and they produce sex cells. So here these are the cells which are being produced. But if you read in detail, we will obviously, we are going to study it in further chapters, that in reproduction, so the female ovary contains all the egg cells that she will produce throughout her lifetime at the embryonic stage only, right? So no egg cells are being produced after the birth. So they are produced inside, when that girl child is in the womb of her mother. Okay, so now there are some cases when the cell goes on dividing and dividing and dividing. It means there is uncontrolled growth. So in those cases, there are the chances that the person may get tumors. Now these tumors can be cancerous, they cannot be, they can be benign, they can be malignant, right? So benign means they will confine to that particular area, but malignant means that these cells will just go into the another parts of the body, and they will cause uncontrolled cell division in those areas also. So this is how what happens to the cells at different places, even in our body, in plants also. So it is not true that the cells, they keep on dividing endlessly, right? Now is the time to discuss a few questions. Question number 1: what is difference between chromatin fiber and chromosome? So we discussed it in so much of detail, right? So what is a chromatin fiber? So can you see this bowl of Maggie? It looks like a bowl of Maggie noodles, right? So I might have put some green color in this, so that is why the yellow noodles have become green in color. So this is what, this is basically the chromatin. This is the nucleus, These are the walls of the nucleus, nuclear envelope. These are the nuclear pores, right? And this Maggie noodle type of thing is chromatin. So it is basically coiled and uncondensed form of chromosomes. Now coming on chromosomes, so what are these chromosomes? Now if you see this is a chromosome. So this chromosome is a condensed form of DNA which is visible only during cell division. When the cell is not dividing, it will look something like this, and when the cell is dividing, it will look like this, right? So I hope this is clear. Now the next question, what are the rungs of DNA ladder made of? So what are those rungs? So just let us make a ladder. Okay, these are the sides and these are the rungs. Now what are these rungs made of? What are these steps made of? So we know there are the three components. What are these three components? There is a phosphate, there is a sugar, and there are nitrogenous bases, right? Now these two forms the sides of this ladder. This means the sides, the strands, right? These strands, okay? And when we talk about the rungs, the steps, so these two are joined, to each other with the help of what? With the help of nitrogenous bases. So say if this is adenine, this is thymine, right? So that ways, if this is say cytosine, this will be guanine, right? So these rungs are made up of nitrogenous bases, so either purines or pyrimidines. So what are purines? So we know there are four nitrogenous bases in DNA: adenine, thymine, guanine, cytosine. So purines are adenine and guanine, pyrimidines are cytosine and thymine. So when we will study the structure of these purines and pyrimidines, that is the time when we will find out what are the difference. So one is made up of a single ring and another is made up of two rings. There are two rings, okay? So I hope this question is clear. Now the question number 3, fill in the blanks. So chromatin fibers are made up of DNA and, so what is the other thing? The other thing is histone proteins, right? So basically the proteins. Okay, now the second one, the two sister chromatids of a chromosomes are attached to each other at. So we are talking about two sister chromatids. So this is the one, this is the second, and they are attached at a one point. And what do we call this point? We call it as centromere. Now this third part, the structure of DNA was first discovered by, who was the first to discover the structure? The first one was Franklin. So she was the one to discover the structure of DNA for the first time, and in the same year then Watson and Crick started making the structure of the DNA. Fourth, the three structural components of a nucleotide are. What are the three structural components? So there is a phosphate group, right? There is a phosphate group. There is pentose sugar. And what is the last one? The last one is nitrogenous bases, right? Okay, the fifth one, DNA strand wound around a histone octamer forms a complex called as. So what is this complex called as? This is called as nucleosome. Now the sixth one, a specific sequence of a nucleotide on a chromosome constitute a specific sequence of nucleotide. What do we call it as? We call it as gene, right? So I hope these were simple, these were easy, because we already discussed the chapter in a great detail. Talking about question number 4, given below is a schematic diagram of a portion of DNA. So how many strands are shown in the diagram? So how many strands are there? So first, what is it? This is a phosphate group. This is a pentose sugar, and these are what? These are the nitrogenous bases, right? Now how many strands are shown? So this is the strand 1 and this one is the strand 2. So there are 2 strands. Now coming on to the B part, how many nucleotides have been shown in each strand? So how many nucleotides are there? So what is a nucleotide? So nucleotide is basically a phosphate group, a sugar and a base. So if we talk about this, so this is one nucleotide base in this strand and this is the second one. And in the same way, this will make one nucleotide and this will make another nucleotide. So how many nucleotides? So there are 2 on each strand, right? Basically there are many, but in this picture there are 2 on each strand. Now the C part, name the parts numbered 1, 2, 3, 4 and 5 respectively. So what is ones? Just now we labeled it, it is a phosphate group, right? Now what is 2? So what is it? It is a pentose sugar. What is this? This is a nitrogenous, I write it over here, nitrogenous base, right? And what is it? The fifth one is also a nitrogenous base. And what is this for? This empty white one. So the 2 nitrogenous bases, they are joined to each other with the help of a bond. So which bond is that? That is a hydrogen bond, right? So we have marked everything. Now the D part, name the DNA unit constituted by part 1, 2 and 3 respectively. So if we just talk about this phosphate group, this pentose sugar and this nitrogenous base, so how many DNA unit, so name the DNA unit constituted by part 1, 2 and 3. What is this unit called? What we will call these 3 combined? So we will call them as nucleotide, right? What is it? It is a nucleotide. I hope whatever we have discussed in this video is clear to you. Now the rest of the portion we are going to discuss in the next video. Until then, keep revising the concepts and make your concepts crystal clear with LearnoHub. Until then, remember, LearnoHub free है पर best है.